Okay, good morning. Let's have a class. And we will continue to talk about DNA replication and then try to finish it today. So let's do some basic review what we did. We talked about the central dogma. Central dogma is DNA replication. And then we go to the RNA transcription. And finally is translation. So that's translation. So we finished talking about here the outline. And then we mentioned several important experiments, which is 1928 F. Griffiths, the Streptococcus pneumoniae study. This study, which tells you the principles of transformation, and which means it tells you there is some of the chemicals there transferring the genetic information. And then in 1943, Avery, McCarthy, and McLean, their group in Rockefeller University, they tells you this transformation transmitter, we can say, it is DNA. Okay, then we talk about is the DNA structure. And I said the DNA is a very boring chemical because there is a major three structures: A, T, G, C is a gene base, the 5-carbon deoxyribose, and we also have a triphosphate. So that's only 3 chemicals for the DNA structure. So we want to talk about something more later on. Okay, this is the time. We move from 1928 to 1943, and right now we move to 1952. We want to go over these experiments because lots of them related to microorganisms. This is another very important research, is Hershey-Chase experiment. This is not a chocolate candy bar. It's not Hershey in Pennsylvania. This is not Chase Bank. That is Alfred Hershey and Martha Chase. They did a very cool experiment in 1952. Alfred Hershey is still there. He later on became a professor in University of Oregon, continued to do the biochemistry study. That is back in 1952. They did a study from a different standpoint to verify the transformation, genetics, the information is DNA. So how do they do? F. Griffiths did it in the mice. We know that. And Avery, McCarthy, McLean did it in humans without animals. What he did is in a bacteria, what they did is in bacteriophage. So what is a bacteriophage? This means they are eating bacteria, live organism. What is that? It is actually a virus. And luckily, by the time they have a virus which is called T2 bacteriophage, and this T2 bacteriophage is a warm phage. So what means a warm phage? There is 2 types of the phage. Some phage could be lysis, which means killing the microorganism, killing the bacteria, and some phage will be integrated their DNA into a bacterial genome. Okay, so that's why we call it a warm phage. So let's talk about what is the structure of phage. This is a basic structure of a bacteriophage. Like this. Bacteriophage is very simple. There's only 2 major chemical components. which is outside here is called capices. The major components is protein. And inside, you will see there is a genetic information there. There could be either DNA or RNA, but never both. And luckily, when they choose this T2 phage, this phage is the DNA phage, which means their center is DNA. And how the phages survive? They need to find the bacteria. This is the bacteria. And in their experiment, it is E. coli. It is E. coli cell. So what happened is, this is the E. coli whole genome. Instead of you killing the E. coli, this DNA will be integrated into the bacteria E. coli genome, and of course, a new phage will be created later on, use bacteria to generate their own machinery. And this is kind of situation we call like a parasite, which means they need a host. The bacteria phage must need a host to survive. Okay? Must need a host. And the host here is a bacteria. And the parasite is a phage. Okay, so how they do these things? Since you have something going to transfer, go to the bacteria E. coli genome, we need to verify this is DNA, not RNA. So first of all, why this comes out with this bacteriophage virus? Now, by the time moved to 1952, just right after the Second World War, they still suffering from the war, but at the same time, they're still doing the research for the Spanish pandemic or Spanish flu pandemic. So there is some idea said, can we just eat a bunch of the liquid with virus, and then the virus could kill bacteria? And I tell you one thing, by the time somebody did it, but it's not very successful. Later on, the phage is purified, it's kind of successful. And right now, they use a lot in the veterinary science area doing the phage therapy. There are some commercial products for that. Although in the human being, in the medical area, they're not doing too much for the human being. But I just want to tell you, the idea by the time, they're trying to eat a solution of a bacterial phage and trying to curing the bacteria. And another thing we also want to mention, by the time in 1952, there is no widely developed antibiotics yet. Or it's still slowly growing. So that's why this is a hot topic by the time. So how are we going to do it? How are we going to verify it? Those two guys noticed that there are very special chemicals belonging to protein and belonging to DNA, but not for the other. So what are the chemicals only existing for the protein, not in the DNA? It is sulfide, is that right? So they could label the protein of a bacteriophage is S35. And which chemical is a DNA but not in the protein? It is phosphate. And they could label this as phosphate 32. It's for the DNA phage. Now, once you see these type of the chemical, S35 and P32, what this means, it is a radioactivated isotope. So you will know the signals there for these isotope chemicals. Okay, so how they do it? They had a bacteriophage. Let's say right here, bacteriophage. Okay. This bacteriophage, I'm going to be growing at S35 solution. Okay, which means we'll label protein. And I have another set of the bacteriophage. What we're going to do, we're going to label the phosphate, P32, we call it the P32 solution, we actually label the DNA. And they both trying to work for a bacteria, which means they attach to a bacteria. They both gonna do that. Once they're doing that, what's gonna happen? Their genome will be integrated. And there is a new phage will be generated. A new phage will be generated right here. But how do we differentiate that? What they do, they're using a big tube. At the beginning, what they do, they're using a kitchen aid blender. They blend it, they spin it up, and then they do the centrifuge. Now, why they can do that? Because the virus is usually less molecular weight compared to the protein. So once you do blender, you're going to separate the virus and the bacteria, and then you centrifuge. What happens? On the top area, you find it is actually a virus. On the bottom area, what you find? It is a bacteria, protein. Then what they find? They find the majority of that on the top. This here is P32, showed up in the bacteria, the bacterial DNA.